Okay, thanks everyone. Just welcome to everyone who's online joining us for this celebration of National Science Week. Welcome to everyone who's in the room with us here at ARI Heidelberg, and thanks for attending. I think we've probably got some more people who will be wandering in as we go. For those who don't know me, my name's James Todd. I'm the Director of Arthur Rylah Institute and Chief Biodiversity Officer at DEECA, so welcome to everyone and thank you for attending. The theme for, or the school theme for National Science Week in 2024 is species survival more than just sustainability. So the theme aims to highlight the importance of science and innovation in ensuring the survival of and thriving of different species in an ever-changing world. So that theme, I think, beautifully encapsulates the critically important role of ARI in providing science and advice to government to inform policies and programs that aim to ensure the survival of Victoria's native flora and fauna species and serve the citizens of Victoria. I'd like to start with acknowledging the traditional owners on the land which we're meeting today. So here in Heidelberg we're on Wurundjeri country, and I'd like to pay my respects to elders past and present and to acknowledge and pay my respects to traditional owners on the land which people are variously dialling in from online. It is gulling season, so for the Wurundjeri, which is that period between deep winter and the full blossoming of spring, and I think with the weather now having hit 20 degrees and the north wind blowing today, that's kind of typical of this season. Gulling is the Wurundjeri word for orchids, so they're starting to flower. There's the wattles are coming into blossom. The lyrebirds are finishing their kind of courtship displays. Koalas are starting their mating rituals with males bellowing, so that's what typifies this time of the year, and it's certainly nice to be out of, hopefully, the deep winter recess that we've been in. So just to go back a little bit about where it all began for ARI. So the Institute here was opened in 1970 by Queen Elizabeth II, and there is a plaque that commemorates that, and for those people who in the room and online will no doubt see that later on. So it was named after Sir Arthur Gordon Rylah, who was a member of the government at that time, of the Balti Liberal government. And I think it's interesting that the quote from the Queen highlighted the need for a government institute for environmental research, because an understanding of the environment and of what contributes to it, what changes it, and of what it is found in it is essential to the management and conservation of the resource. So I think it was an interesting time for, I think, environmental awareness at that time in the world. Rachel Carson's Silent Spring had been published in the 1960s, which had brought about a greater awareness of humans' impacts on the natural environment. But equally, around the same time, the Victorian government established the Land Conservation Council in 1970, which was in response to a rather controversial decision to clear the Little Desert and turn it into agriculture, and it was probably one of the first big environmental movements, community-led movements, in opposition to that idea, and so that came out of that process. So I think fair to say that the establishment of ARI was at a time when there was growing awareness about the importance of the environment and the science that underpins it. Welcome. Now this is a little overview of some of the work that happens at ARI. So we're based here at Heidelberg at 123 Brown Street, and we've got a building that's probably got about room for about 110 staff. And we've got about 90 people at the moment, and we have regional staff here as well from Port Phillip but other parts of DEECA. And this is just a little video of the various work that goes on in terms of scientists in the field, surveying for things like crayfish, looking at cetacean identification, migratory birds, etc., and forest surveys. We do work on fish to work out what their age is through analysing the bones of fish. There's leadbeater's possum. There's a whole heap of stuff around, I assume that's something like mud islands, where we're looking at nesting birds, including migratory species and the like. So the breadth of the work that we do is huge, and I'll touch on that in a little bit as part of this presentation. So ARI today, so I think it's probably fair to say that 50 years ago the Institute was probably founded on the basis of interests in hunting and fishing and the wildlife aspects that go with that. Since then our skills and capabilities have evolved to meet the changing needs and demands of government and the Victorian community. We're now equipped with the skills and capabilities to answer complex environmental questions with the latest technology and in partnership with other experts and stakeholders. While we do still collect vital records and observations in the field, we are increasingly involved in large data analysis and modelling and interpretation to inform decision-making processes in an increasingly complex policy environment. So I think this sees our approach to this means that the environmental research that we conduct here at Rylah underpins government decisions, policy, regulatory policy, investment decisions, planning and the like, both here in Victoria, but now also increasingly internationally as well and in other states. Our demonstrated reputation and scientific credibility means that we can deliver science and research that supports evidence-based decisions by government and other land and water managers, and it helps provide answers to complex policy problems, as I've said, and it targets, monitors and improves environmental management actions in a changing climate. There are very few environmental institutes like ARI remaining in the country. I think most of them have actually disappeared, certainly within the government sector. So it has demonstrated its longevity, but also the fact that its support through the Victorian government and the long-term commitment to robust evidence underpinning policy development in the state. I consider that ARI is uniquely positioned because it is a part of government. It means that we can be trusted. We are colleagues with policy developers, and we work under the code of the Victorian Public Service, so we can deal with highly sensitive matters when we're working on those particular issues. So I think the important bit here is that we foster our long-term partnerships with policy and other resource managers. It's not only just the what and the why, but also the how we go about it. So the partnerships are really important. As I said, we understand the policy development process and the policy environment and the role that science has in informing that. And we work with our policy partners to develop the necessary science to support decision making. I think working hand in hand and in partnership with our policy colleagues is really part of the unique ARI value proposition. Equally, we're embedded in the Victorian State Government, and as I said, part of that comes with the VPS Code of Conduct, and we can be trusted. We pride ourselves on integrity and quality and adopting, of course, and the values and behaviours that come with that. I think our people are our greatest asset. We have really diverse species and ecosystem knowledge. We develop innovative approaches and apply cutting-edge technology and are routinely required to analyse large data sets drawing on more than 50 years of ecological data from across Victoria. And we continue to attract and retain the best scientific minds with a broad-ranging skill set, and we can bring those capabilities together from across the Institute and indeed across the broader academic and research sector where needed to help answer increasingly complex policy problems and respond quickly to urgent requests or emergencies as they arise, or emerging issues as they arise. And we're continually focusing on the science capabilities that are needed to support government both now and into the future, and building on those capabilities where we need to. So this is just a little snapshot of the kind of the partners that we work with. So at the moment we've got over 50 clients and collaborative partners from a range of different organisations from across the sector, government, so obviously within DEECA itself, but across government, statutory authorities, traditional owners, other research institutes and the like. We're about $25 million business per annum, and over 95% of our work is fee for service directly to a range of clients, including those groups that are sitting within the DEECA itself. So Bushfire Forest Services, obviously Biodiversity Division and Water and Catchments being the main contributors there, as well as the regions to some extent. And also means that we are accountable to our clients and the feedback that they provide. So In 23-24, we delivered over 780 odd milestones for over 300 projects. So it kind of gives you a sense of the volume of the work that goes on here. But pleasingly, we had a feedback from our client surveys said that we were rated as 100% by those clients as good or better, with 86% saying it was very good or excellent. So we continue to look for opportunities to improve in that space where we can. It also means that this diverse funding portfolio means that we can leverage off some of this investment to help address broader government needs. And so sitting within government, this is our structure currently, of course. We're sitting within Regions Environment, Climate Action and First Peoples within DEECA as part of Biodiversity Division. And we're part of, so ARI is now part of a new branch which is called Biodiversity Research and Information, which includes Arthur Rylah Institute and the sections that sit within it, but also includes the Biodiversity Information Systems. So the fact that we're embedded with government, and as I said, we form partnerships with policy and program colleagues, and we're very much attuned to the government needs, but that leverage of external projects means that we can bring that information and that knowledge that's created through those projects back within DEECA's decision-making processes and within the Victorian government making processes. So there's a lot of value add essentially to that work. I've talked about our people and how important they are. At the moment, we're about 80 plus scientists across a broad range of scientific fields, which are highlighted on this slide. I won't go into too much detail about those. Currently, this is our senior leadership team, and you'll be able to see some of those or meet some of those people today for those who are here in person, as well as potentially for those who are online. But we're set up around myself as the ARI Director and Chief Biodiversity Officer. We have areas of scientific endeavour attached to applied aquatic ecology, wildlife ecology, community ecology, and then an area around knowledges, behaviour and science impact. And then, of course, there's the business and admin function which supports the operation of the Institute and making sure that we're meeting all our regulatory and reporting requirements and safety requirements and the like. And there's a couple of positions that are currently vacant that we're looking to fill in the near term. So just to give you then a snapshot of some of the more specific areas that we're focusing on and that we implement. One of the big areas of scientific research and endeavour in the Institute is monitoring and modelling. So we use a range of high-tech equipment and processes to do things like remote sensing, as well as things like call recognition software. So we've developed some of that in-house to help us when we're putting out audio recorders into the environment. We can use machines to identify what species are calling in those environments, a much more effective and efficient way of trying to collect biodiversity data. We have models that help track wildlife movements, as well as models around developing things like population viability models so that we can start to understand the impact of management on persistence of species. And more broadly, we do a lot of spatial modelling of all Victoria's native plants and animals, over 4,500 habitat distribution models, but also increasingly and importantly, it's about also modelling the threats to those species and what the benefits of actions might be to mitigate those threats. So we bring that together in a way to support government decisions, regulatory settings, investment decisions and the like. We also have a range of programs that are actively working to protect species, particularly threatened species. We're leading on the 10 in 10 program, which is a program aimed at bringing 10 aquatic species back from the brink of extinction, and a key component of that is the management and coordination of the newly opened native fish conservation hatchery at Snobs Creek near Eildon. We input into a range of things around captive breeding, and you'll see some of the tanks today on display which have a number of threatened freshwater species in them. We have a threatened species genetic rescue research, so this is where we're looking at the science of genetic decline and how we can better recover species through things like gene mixing and the like. We're doing a lot of work in the bushfire and flood response and recovery, and increasingly thinking about how we can better prepare for those emergencies going forward. And lots of work around habitat restoration, invasive species control, those sort of activities. And then in thinking about how we need to adapt for the future, and particularly what the needs of government, there's a range of research areas that are starting to come to the fore, which we're prioritising. We've got a big program around transition to renewable energies and how we support that through research and spatial modelling. I've mentioned bushfire and flood preparedness and response, but equally we're starting to think about how can we identify the risks to biodiversity from future fire, and working with our colleagues across the department in modelling that, and therefore what sort of actions might we be able to undertake to help mitigate that into the future. There's a large program of work with Water and Catchments Group in the department around water for the environment, modelling the impacts of environmental water flows, plus a range of other water-based activities. A new area that we've really just got into, or starting to get into with our Ag colleagues, and Angela's here somewhere today, is around soil health. So we're starting to look at the soil kind of biodiversity essentially, which is probably orders of magnitude greater than the above ground biodiversity. So it's starting to really develop some really neat technologies to sample that, but working with Ag across natural and productive landscapes to understand that better. A big area for us is also around participating in two ways of knowing. So this is the idea that how do we bring Western science and traditional owner or traditional science and knowledge together to support decision making. And we've got a number of projects where we're working with traditional owners, whether it's working on eels with Amara, or whether it's freshwater crays and turtles with Yorta Yorta, or whether it's cultural burning with the water, or forest management with Jara and a range of other TOs. There's lots of projects going on in that space, so it's a really exciting and emerging area of endeavour. And probably one which we've started some work on, and again supported from biosecurity area within AgVic, is starting to look at wildlife disease preparedness. And of course we've got a looming H5N1 risk in terms of this spring, in terms of impacts on wildlife. So some of that research is being pushed straight into action in terms of how we prepare and potentially respond to that virus should it arrive. So in summary, thank you again all for attending, both online and in person here. Here at Arthur Rylah Institute, we are delivering critical science that is helping address wicked policy problems, and how we bring the capability from across the Institute together to address those is really one key part of the value proposition. We are uniquely placed within government to provide trusted science and with the capabilities needed for the future, and we'll continue to invest in the capabilities that we think government will need in the future. And we have a very strong focus on partnerships, both internal to DEECA and within the division of course, but also external clients, and wherever possible we're looking to leverage that investment and value add to that to support better decision making by governments in relation to biodiversity protection and management. I might leave it there, so thank you. I think Andy's going to wander off now with the laptop to start the online tour, and I think for people in the room you're going to stay here briefly, and I'm happy to take a few questions from inside the room, but then we'll get organised and get our tour underway. So thanks all for attending, and good luck with the online tour, guys.
Thank you so much, James. So we're going to take this laptop away with the 120 or so people online and leave you guys for your own private discussions. This is a new sort of way of doing tours at ARI. We are always innovative in the way we communicate our work, and sometimes it works, sometimes it doesn't. So wish us luck. Tess is going to be our director of... Oh yeah, here we go. This is going to work. All right, wonderful. We'll catch you guys later. All right, see ya. Online, is that working, Tess?
Just turning on the audio.
All right, see how we go. See you guys.
So I did welcome you both, and I welcome Nick at the start. So whether you guys...
Yes.
Yes? Okay, wonderful. All right, so we're here in the beautiful foyer of ARI. Do you want to let Joseph, can you go? So this is how most people enter the building. You'll be lucky enough to be our reception. Maria's here today, and she's also standing by some beautiful artwork, which you'll notice most of the times you come to ARI. We have a lot of great traditional owner inspired or designed sort of artworks. And if you come over this way, I'm going to start this sort of on the in-person tour with a bit of a view of the striped legless lizard. So this is pretty much where everyone starts their tour, standing here and playing Where's Wally. His name is not Wally, but it's just the most fun part of everyone's day to see whether or not they got the lucky moment to see the striped legless lizard. This one came from Victoria University about 15 years ago. So it's over 20 years old, which is much, so around the sort of life expectancy of striped legless lizards. We did have two, unfortunately lost one, but this one is still happy and strong, and I don't think I can see it today, so you're not part of the lucky viewers today if you get to see it. This is one of our sort of showcases of some of the species that we have worked on in the past. It is a threatened species, so we won't be replacing it in future, but it was a nice thing to be able to help out Victoria University in providing it a home. If you come over here, we've got this amazing bit of artwork by Auntie Kim Wondon. You good?
Just trying to see if I can spotlight for myself.
No worries. So Auntie Kim Wondon worked with ARI and just sort of creating this artwork in collaboration in some ways. So it's all her design. She uses a lot of the native materials or natural materials that show there's a sort of ephemeral part of the nature. But it also includes a really unique part of grass, which talks to the sort of always was and always will be Aboriginal land. So it's a really unique sort of addition to this style of yuck trap, which is designed traditionally to capture eels in nature. So ARI, through a program called Miranagu, was able to contribute and participate in the process and weave in some of their own personal items and objects. So it was a really great experience to be able to build a collective story and show what this meaning, show the sort of joint meaning together. You can also see up here a design by Brendan Kennedy, which talks about the Tati Tati seasonal calendar and the animals that sort of move through.
Andy, we need to turn off the mic in the other room.
Oh, okay. I will be right back. Sorry.
Thank you for people online. Yes. Okay, well, the artwork I'm pointing the camera at right now is a Brendan Kennedy piece that was created to depict the seasonal calendar from the Tati Tati traditional owner group. And Andy's back. Okay,
all right,
cool.
We're going to continue the tour. Hopefully some of that was audible and you can sort of continue. Over here, we've got a kangaroo taxidermy from Melbourne University. So this was a gift from just last year that was sort of highlighting a 100 kangaroo sort of tracking and monitoring program that started in 2004. Vlad, as he's clearly named on his collar, was someone that they trapped early on, I think in 2011 or something, and monitored him throughout his entire life. Sadly, he was found injured on a golf course and had to be euthanized, but he continues to sort of communicate the research and importance of monitoring kangaroo populations from across Victoria. And that's some work that Melbourne University continue to do to this day. We'll talk a little bit about this. So this is actually one of the artificial tree hollows. So in Victoria, we do have a lack of tree hollows in certain areas, which are really important for a variety of species. Many of the animals like leadbeaters, possums or gliders require a hole that is just the size they can squeeze through themselves. So what we were doing as part of our research was creating these artificial hollows and putting in these pre-built doors to help sort of create these cavities and spaces for leadbeater or animals to nest in. At the time of the finishing of the research, about 50% of the hollows that were created were occupied. So that was a really encouraging result, and it showed that it had sort of created a bit more hollow-bearing habitat for species. Let's continue through here. You get a brief view of the fish tank, but we're going to come back to that with Jared, hoping that the technology holds up, but we'll see how we go. We're going to through this other area and hopefully find a net in the herbarium. So as sort of James alluded to, we've got a really big building, can host about 110 different staff members. At the moment, we're almost at capacity, but it doesn't mean that we don't have a wealth of knowledge existing here. And if we come in through here, welcome to Annette's domain. This is the herbarium. So I'll stand with you so it's not so awkward with the camera. So Annette is going to tell us all about herbarium and I guess why we have one.
Yeah, sure. So this is used by the botanists, so people who study plants here at ARI. First of all, what is a herbarium? People don't know. It's a collection of dried plants like this, dried plant specimens. Why do we do it? We do it because we need to have accurate identification of the plants that we bring back from the field when we're doing our plant survey. So we do a lot of plant and vegetation surveys across the whole state. Sometimes, a lot of the times, we know the plants in the field, but sometimes we need to check them more carefully under the microscope and check them against the definitive descriptions which are made by the Royal Botanic Gardens, which is the statewide authority. So essentially, if we wanted to check on something we brought back from the field like this, we'd put it in a plant press to make sure it kept okay until we got back to the lab. We could then look at this under the microscope, the one we've brought back, and we could also compare that with one of our in our collection here in the cupboards, where the identification has been confirmed. And I'll just show you what's on the computer here, if you can come over. So this is what we're looking at under the microscope. This is the fresh specimen on the underside of the leaves, and this is the old specimen, which was collected in 1978, so it's still really clear. You can see these star-shaped hairs, and this is a diagnostics feature for this particular plant that we're looking at. What we do is we use a key that's the Royal Botanic Gardens has an online key for all the plants in Victoria. How many of them? 7,000 of them or more, and you make your way through the key and you discover the features that are really separate one species from another. In this case, one of the features is these star-shaped hairs. So it's not something you can see with your naked eye. It's not something you can see in a picture or a photograph. And in a description, you want to be sure. You want to be sure that's what you're really looking at. And so that's why the actual specimens help you really confirm that. So in this case, this is a widespread species. But there are ones that are very rare that we look for, and this helps us in the sort of threatened species work we do. So if you think you've found a threatened species or a rare species, you really want to be sure that's what you're looking at, because they're much harder to recognise. And so that's part of it. If we're still not sure, if we still can't determine it from what we've got here, we would of course consult with the Royal Botanic Gardens. So with our collection here, we've got about a quarter of the species or taxa in the state. So we hope as time goes by, opportunistically we add to those collections. So I think that's probably all I need to say about that. Only that it's a really useful tool, and we get particularly busy here in spring when we're coming back from our field trips with plant specimens that we want to check out. So yeah, and all of this it's really important that we're accurate with our identifications, because we need to make decisions about threatened species management, or policy terms, what's happening with fire management and things like that. So that's the reason we've got this as one of our tools that we use in doing our work.
And Annette, some of the people online might know you from your seminar that you did not too long ago.
Thanks.
But in terms of barring all Banksias, what would be your favourite specimen that you've got here in the 2,000 or so species?
Well, now you go.
Don't ask a botanist what their favourite animal or plant is.
Don't have a favourite, no. So no, it's just a wonderful collection that's been built up by people over many, many years. I mean, it does date back to the 70s. People have built on each other's work. It's a very old-fashioned, if you like, technique, but it's still highly relevant today. And I think that's what's interesting. The Botanic Gardens itself has specimens dating right back to the very first, early settlement of Victoria, and And yeah, that's still in use today. So I think that's what it's about. We build on the work of other people.
Yeah.
So,
And I think you mentioned that some of the ex-ARI botanists are still coming and volunteering their time. So once you're involved in a big project like this, it's hard to walk away.
well, it's an important collection and it does need maintenance. So I think that's fantastic that we've had that commitment. Yeah. People? Yeah.
So thank you, Jeff,
Yeah.
and thank you, Judy, if you're watching.
Yeah.
All right.
Thank you.
We'll continue on. Thank you, Annette, so much for your time.
Thanks, Annette.
We're following Josephine and Tim upstairs to the spatial room, which is the next part of our tour. Also, you might notice as we walk through some of these beautiful artworks on the walls, they were donated by, I think it was Arthur and maybe Samantha Howard. Carolyn Howard, back in 1970, wasn't it? With the opening.
Yes, it was.
Yeah. So we've got some beautiful
Several hundred of those prints around the building.
Yeah, I think someone said it was 400 different artworks throughout the building, all nature or natural sort of prints, which is really quite an asset to the building.
And don't fall down the stairs looking at them, Tess, please. Be a fail for the technology.
Yeah, well, I think we're just holding up. I think the internet's still like getting an upgrade. Right. So we are currently in the spatial lab on level 1 of ARI. Tim, what have you got here to show us?
I've got you lots of computers here to show you, which is unfortunate because spatial labs is mostly about analysing data, and that's what we largely do in here. So we've got some pretty reasonably high-powered computers. In fact, this one here is processing 5 terabytes of koala calls from the Mornington Peninsula at the moment. So it's probably several months worth of data when we're looking for koalas in different locations around the peninsula. So that computer will just plug away, listening away, rather than someone with headphones. Saves a whole lot of time and effort. It's a really efficient method that we've developed here. So the rest of it is computers. So we use these computers to generate information for biodiversity management, basically. So a whole lot of, as James's overviews mentioned, we use it for biomanagement, water management, threatened species management, land, even just general land management decisions like native veg regulations and things like that. We built more than 4,500 species distribution models. They underpin a lot of what we do here and as part of the lab's work here. So species models are used for decision tools around where should we do fox control, or where should we do weed control, or where should we burn more often or less often, or add water to, or which areas really need protection because they're highly valuable or highly rare threatened species might exist there. So we build a whole lot of those tools here. Someone might have a bespoke question around what do we do for best management action for this small area of park, for example, or it might be at a statewide level. So the decisions are made at that broad range of scales as well. So we largely build statewide models, but we do also build bespoke custom ones for smaller areas as well.
And you've got some pretty great props here on the table here. What are we looking at?
What are you looking at? You shouldn't ask me this.
Nah.
I know what they're called. So these
Josephine's in the wing for any tricky answers, I'm pretty sure, to help out.
So that you can see this one's got a microphone on it, and these have got microphones in them. This is a mini song meter. So we put these out in the field rather than we might do a bird survey for 20 minutes and you go and hear what birds are there. These things can go out for months and months at a time, and they'll just keep recording the noises they hear. So you can bring them back, and historically someone would just listen to that with a pair of headphones on and say, that's a magpie, really exciting, or that's something else. What we do now is we download the data, we train the computer to recognise the call for a certain species. It might be a bird, a bat, or some sort of mammal.
Yep.
And we plug the data into the computer, and then that listens for us. So it can do like 400, it can do a year's worth of calls from one of these in 36 hours.
Wow.
And it's pretty torturous if you're trying to listen to that many calls by headphone.
Yeah.
So there's a couple of different types here, but we would recommend this type. These are audio moths as well. They're a bit smaller, a bit more compact and a bit cheaper, but these ones here seem to work really well.
So you're definitely embracing the idea of work smarter, not harder.
Work smarter, not harder, but it's also a snapshot because we might record something today. We might not have a recogniser for all the species that are there. We can go back and reanalyse that data and find more species. So we've currently got about 50 species we can identify by their calls. But if that's 400 species in 10 years' time, we can go back and run all this data again and say what else was there at the time. So it's great legacy data as well.
It's really cool.
Yeah.
And I'm going to rope Josephine into this next question. So Josephine works in decision sciences. Was there anything you want to sort of highlight about the skills and capabilities of your awesome team?
Well, I think for me, decision sciences is a really exciting area of research because it is able to bring data and models into a decision context to make it more kind of easier to sort of navigate. Like, what decision do you make? Sometimes when you're navigating a decision, you've got multiple values. Biodiversity is not the only value you might be considering. It might be you're trying to protect life and property, or you're trying to support recreational users of an area. So decision science, I think, is such a fantastic tool that separately acknowledges the data and models that support recognition of those values, and then puts it into a framework where you can really understand how the decision that you make really benefits or impacts on those different values. So yeah, I think it's a really great way of unpacking messy problems and putting some structure around it, adding transparency, involving a whole bunch of different stakeholders who might have different value preferences, and then ensuring the science is really tuned to that particular context in a way that is useful and meaningful.
Totally. And I think that's what's so exciting is like you've got all the people collecting the data, then you're analyzing it all, and then you're helping make decisions, or people make the right decisions and the things that they value. And it's like an end-to-end sort of process, which we've got the expertise and capability for.
Yeah.
And we've got 85 or 90 scientists in the institute. We've got a broad range of expertise. So if you want to know something about any particular species, we've got someone to ask, including, people who understand what's in the soil now. So,
Yeah.
Which is scary, the numbers, as James mentioned. So.
Yeah, exactly. Well, thank you so much for your time. Is there anything else you want to add before we run to meet Gemma downstairs?
No.
No? Perfect.
Have fun in the lab.
Thank you very much.
Cool.
Bye.
Show you some of the maps.
Oh, you're looking at maps?
Yep.
Oh, did you want to do a collapse on the pretty colours of the maps? If you want them printed for your own personal walls, talk to us later.
Call
So we're going to go back downstairs.
Sorry for all the motion, everyone.
Yeah, people don't get motion sick, but we'll check the feedback later on how many people felt motion sick. But I think you're doing a very good job, Tess, on the computer, walking around with it. All right. So we're just coming into the library now, and if you look out the window here, you will see a bit of the internal courtyard areas. So we actually have Murray River turtles and eastern long-necked turtles in there. One of them is called Kevin, or Footloose. He lost his leg to what we believe was a eel that was also living in the courtyard at one point. So he became Footloose because he was lost the foot. But I also like to think of him as Kevin Bacon because he also was bacon at some point for some animal, which we think is the eel. Then we have, I'm just going to point this one out too. So this may not look like something online, but you can see like it is a print of an emu. We have all of our meeting rooms and things sort of decorated with a stylized image of photos that our researchers have taken from the field. This one was a Steve Platt's photo of emus out Western Victoria. So it's the really nice sort of part where all our staff contribute to the way we sort of design and lay out our building, but also just the feeling of us being a big group that works together. In here, we're walking past the wall of all of our current or last year's achievements for publications. It's a good way to celebrate all the work we do. And we also have a library, which is a bit of a remnant library from the days where ARI didn't have any sort of online internet or anything else like that. So all this used to be like the core knowledge set. It is a lot smaller now because a lot of things can be found online, but this now homes a lot of the resources that we can't find online or anywhere else. So it's a really important physical copy that we have record of. And we also keep a record of a bunch of journals in these sort of compact sort of areas, which is a really nice way to remember all the different pieces of work that ARI has contributed to over the years. And in this room, we're coming up to Gemma. Gemma works in the wildlife ecology team. What have you got here, Gemma? This looks like a lot of cool stuff.
Yeah, hi everyone. I'm here today to talk about wildlife ecology and the work we do. I've gotten out a lot of different gear that we use when we're in the field. So sometimes we're out there trapping. We might use things like these cage traps to catch medium-sized mammals. When we're out looking for really small marsupials, We often use these Elliott traps, which are a lot smaller and darker, and you can squish them in more dense vegetation. I guess a lot of what we do is collecting field data to inform our science and our questions that we might be asking, working with collaborators and clients to ask. So we'll often work to come up with some really robust study design. We'll go out and collect the empirical field data. And wildlife ecology, we're really lucky. We've got a fantastic group of statisticians in our team that collect a lot of do a lot of quantitative modelling. So they work with us as scientists at ARI, or they'll work with other parts of the department to help do something really amazing with that data so that we can answer those questions. So they might help us do some modelling or statistical analysis to do that work. So some other equipment that we use, I've brought in today one of our taxidermied leadbeater's possums. I don't know how close I can get, but some of our team here at ARI have worked on leadbeater's possums a lot over the last kind of decade. It's a really special species in Victoria. It's only found in Victoria. It's a critically endangered species. And so our team here have done a lot of work to improve our survey methods around that species, working with other organisations such as Zoos Victoria. So we will use things like this thermal imaging camera, which picks up heat signatures. And so we can go into the forest. It might be a bit hard to see, but we can go into the forest and look around in the trees for anywhere. So anywhere showing on here that's red is sort of a hot area. It's not the best inside, but in the forest, it means we can try to find these really tiny possums that are running around and they're moving really fast in really dense vegetation. So we might use a thermal camera, or we've also developed some techniques using camera traps.
I'm going to stop you there for a second, because if you really want to see how the thermal camera works in virtual reality, join us at Nicholson Street on Wednesday for the VR, or you can find it on YouTube. There's a great little video of me and Jem out in the forest looking for them with the little thermal imaging thing. So
great,
cool.
Great plug.
Yeah, good plug. Yeah. I'll let you keep going.
Yeah, we've got these camera traps here. They've undergone some different designs, but basically we can put batteries in these and use these to remotely detect animals. So we often work with arborists to climb up the trees and put these cameras up in the canopy so that we can record possums and other species. And then we'll come back and collect them about a month later and look at all the images. So that's also been working really well. Some of the other work we do with other wildlife in our team uses telemetry. So we might put trackers on things like turtles. So this is a broad-tailed turtle here that I've put out. One of my colleagues, Katie Howard, is doing a great project at the moment with the Yorta traditional owners up in Barmah, and they've put acoustic devices on these turtles. They can track them. We use a little tracker like this blue one near Andy to track terrestrial species, but because turtles live underwater, it's a little bit different. They actually have a cool array of acoustic monitors through the waterways that they also use for fish research. And so that every time a turtle swims past there, it'll take a little ping of which turtle it is, and then they can go back and go out in the boat and download that data and look at movement and stuff.
And there was an awesome article when this research started, because it was quite expensive originally, and the journalists called them turtles get Gucci backpacks or something like that, because the cost of some of the backpacks were equivalent to Gucci bags, which was a really great way to sort of share how important, but also how innovative that work was.
Yeah. So that's ongoing at the moment, which is pretty cool. So yeah, we use heaps of different tech, I guess, in what we do. We've got lots of people that head out into the field, and then lots of great capacity here back at ARI to help analyse that data and provide information to decision makers about how to use it. I reckon that's about it.
Yeah, awesome.
Yeah.
Thank you so much, Gemma. Really appreciate it. And if you didn't get a chance, you can see some of the cool books that they use to help ID things.
Yeah, we use all of these field books pretty routinely in our work. Species like microbats, we've got some great capacity here at ARI with Lindy Lumsden and her team. We often do microbat stuff, and microbats are pretty hard to ID, so it's really handy having Lindy's brain, but also some really good field guides with really good pictures and descriptions of the different species.
Awesome. Thank you so much, Gemma. That's wonderful. And I love all the props.
Yeah.
Props to the props.
Love a prop.
Yeah. All right.
See ya.
I think for the next bit of tour, we may have overlapped with the in-person group. So what we're going to try to do is get you out to the boat shed and see some of the cool sort of vehicles and utility that we have. If we do drop out, it's because our Wi-Fi isn't ideal in all spots. So if that happens, we can try and come back a bit later for the tour with Jared, who will be talking about, I guess, the aquarium, the work that we've done in sort of bushfire recovery with the fish, and also the 10 and 10 program, where we're trying to shift the conservation status of 10 aquatic species to less vulnerable categories. So, do you reckon we go this way? Do you reckon that's a good idea? Do you test?
Yeah.
Okay,
Yep.
we'll try that. And if not, if we drop out, we'll be back in at least 5 minutes, and we'll go straight to the aquarium tank inside in the foyer.
Sorry about our squeaky shoes, everyone, if you can hear them.
Well, actually, I'm going to take you to a different spot first, because this is a piece of communication stuff that Tess worked with the aquatic team on. We're going to go this way first. We're just going to bring the online tour just briefly to see the. Where's all the transmitter things? Wasn't there another one?
We've got it waiting out in the shed.
I was waiting in the shed. Oh, sorry. I was spoiling the surprise. We're going to go somewhere else. Thank you. All right, they're all in there. Okay, we'll go to the shed first, and then we'll come back in. So out here, hopefully the video quality is still good. This is where we keep our massive fleet for a lot of the in-field work that we do. As you can see at the moment, it's a mixture between personal and private cars. At the moment, we've got a little bit of field work, mostly in the aquatic space, but it'll start ramping up a little bit more as the weather gets a bit warmer. We have specialty sort of equipment. Yep, keep going. Oh, they're in there. Oh, okay. We're going to go to the fish tank.
Go back inside. Sorry, everyone. We've caught up with the other tour group.
So we had timings on how we're going to run this, and we're doing very well. We're right on schedule. I think they're a bit late, so, you know, they're not keeping up to the time. We're going to go through the courtyard area and see if we can see some turtles along the way. I think it's a nice segue given Gemma's latest sort of update around the broad shell.
It's the tour is turning into a bit of Steve Irwin crocodile / turtle hunting from Andy.
Can't see it. Must be in the reflective bits. You can't see them.
We look at the fish tanks.
Yeah, we'll go to fish tank. So with the fish tanks, and hopefully you can see it quite well. This is a really, really great example of the types of species that our aquatic team are working on through the 10 and 10 Conservation Hatchery, which is also in partnership with a whole range of other groups, including Victoria, the BFA and federal government. This is a showcase, and you might be able to see the little name tags down here. But they talk about a lot of the native fish, and you wouldn't see this combination of species in the wild at all. So this is very much just some of the species that we worked on, and they needed somewhere to be homed. But yeah, it's not a representation of a true sort of ecosystem that you'd find in the wild. But in here, you can see some of the longfish. I like to call them sausage fish, but no one else should. Galaxids of varying types. So Tamo Radic, who's worked on this species for many years, many of you might know him, is an advocate for it and helps sort of rescue lots of different threatened galaxid species and identify which ones are unique. We've also got some of the southern purple gudgeons. I think that's one there. It looks like one. So the southern purple spotted gudgeon is actually a species that was considered extinct in Victoria up until maybe 5 or 6 years ago, when one was found up, I think, around Crank Lakes. I might have that location wrong, so don't quote me on that one. But the southern purple spotted gudgeon was considered a zombie fish by a lot of the media because it sort of came back from the dead. Now we've got a really great breeding program in partnership with the 1010 Conservation centre, but also some really good in-house expertise that helps to build that capability up. We've also got some species that are part of a Glenelg freshwater mussel sort of breeding program. The freshwater mussel has a really interesting life cycle where part of it, when it's sort of after it spawns, all the baby little mussels actually need to attach themselves to fins of fish to be transported up river streams and to different areas. So in our sort of trials, we had some mussels that were about to spawn, and we used some of these fish as some of those surrogate sort of vectors to help work out that life cycle and inform the conservation hatchery approach. So some really, really cool work that this tank really sort of speaks to, but it's also like a great way to introduce guests to the types of work that we're doing. We also have in here grayling, which I think have also been coming back in sort of the southwest of Victoria in recent years, and some really cool shrimp that I don't know if they have any sort of conservation relevance, but they're very cool in here. And when we go to the aquatic lab, you have a chance to see some of the crayfish that we do work on, and Jared will talk in more detail of that. Can you see any close-up ones? The big ones that had the massive arms this morning, Tess?
I think they're all kind of at the back. I don't know. The reflection seems pretty strong on the camera here.
I might be able to. Let me see if this helps. Is that better?
People online, yeah, that looks a little bit better. People online, are you getting a good view of the fish tank? Got a pygmy perch up close there.
They're very curious about this. So for those who miss channel 31 fish channel, like I do, this can be a nice therapeutic 30-second break during the day to reset. If there's a request for online streaming of this, we may make that available for you in future. All right.
Oh, we've also got Murray River rainbows in here.
Yep.
Which are pretty cool.
All right. We'll head to the other tank. There's only 2 fish in the other tank, so it's a little bit less interesting. Over here, you can see the Australian bats. We've got 2 of them in here, and these are quite small, I guess, small versions of what they can be. These Australian bats can grow up to 3.4 kilos, quite a large fish. And they love sort of estuarine environments, but they also tolerate freshwater areas quite a lot too. So this one, I believe, is in a freshwater tank. There's 2 of them, great little example, but yeah, it's nice to see different varieties of fish in the workplace too. What else can we do? Am I hearing voices? We could do, what we're going to do is we're going to do a really quick walkthrough of level 2, just so.
I reckon we just go to the aquarium.
Go to the aquarium? Okay,
Yeah.
we're going to go for it.
Because we're getting close. We're thinking this will probably end about 2.
2 o'clock, yeah.
Sure.
We're running about right on time.
Well, it's nearly 2 now.
Jared's a bit of a talker, so I think he's had them for a little bit longer. I reckon he's back. We're going to be. So this is where most of us don't get access, so we have to knock very politely and hope someone's around. It's not a coded lock, so don't memorize the pattern.
Hello?
Hello. Can we come in? No,
I just want one quiet. Come on in.
Thank you. Whoa. I mean, I'm not surprised by this because I've been here a couple of times, but I'm sure most of you are surprised by this. What are we looking at, Jared?
All right, so g'day everyone. What we've got here is the ARI aquarium. So come in. There's not much happening at the moment. It's a experimental aquarium.
You're good?
Yeah.
All good. So basically, over the years, we've done all sorts of things in here. Everything from held Glenelg mussels for spawning trials. We've held fish that are rescued from bushfires, so the 2020 fires, came back in here, really threatened the laxids from Gippsland or the quarry perch. We also had some held back here. We can hold them in captivity as an ex situ population, a bit like people might remember the bristlebird rescue, same thing but with fish, and then we take them back to the wild after the threat has passed. So generally for fish or crays or mussels, that's after it's rained, so you've had all the inflow of ash and sediment that you get after fires. Like I said, it's also an experimental aquarium. I started here actually a long time ago now, but looking at the impacts of cold water on swimming speeds of fish and relating that back to cold water coming out of large dams. We've recently had some students in here looking at the thermal tolerance of rainbow fish eggs and larvae, and that's important again because of the way we now deliver water for the environment. We need to make sure not that we just get the flow regime right, but we get the water quality right as well. So it's all well and good delivering a flow, but if it's too cold or too hot and you don't achieve the aim that flow is targeted for, it's not an ideal use of water.
Hard to explain.
We also hold recently had some purple spotted gudgeons in here as part of the breeding program for that species to then get liberated out into the wild. What else can I tell you? We're about to undertake a partnership with the Trove Uni, who will be coming in here for a couple of years and doing some experiments on marine species. Looking at a range of things on flathead and bream, which will be really exciting. But I suppose it's really well set up for experiments. So each of these tanks, which you might be able to see, we can run different temperatures in each of the tanks, different flow regimes, and we do.
Yeah.
That's what we do in here. So
I think I remember the Murray Hardy had. You had salinity trials,
Salinity
and things
trials,
like that, trying to work out like what the tolerance is, especially with whether they need environmental water to keep the populations alive and stuff.
that's right. Yep, and Murray Todd with salinity trials many years ago now. So yeah, it's a great facility. We've done the breeding trials for the Glenelg mussel. So some people might know about the Glenelg mussel. Critically endangered from the Glenelg system, of course, but a couple of spots in down the southwest. So we were actively able to get those to spawn in captivity. They've got a really fascinating life cycle. They produce a little strand of gelatinous babies, which are called Glycidia. And those Glycidia, that strand waves like this in the water, and in the wild, fish think it's a worm, and the fish will come in to eat that worm. Those Glycidia then attach to the gills or the fins of the fish. That's part of the life cycle that needs to happen for about a month. They then drop off into the sediment, into the sand and grow. If that fish isn't there at the right time to come and sort of attack that worm, then the life cycle doesn't continue. So obviously having the right mix of fish and mussels there at the same time is really important for that species, and probably one of the reasons why it's threatened. That species now, we've moved up to the new conservation hatchery at Snobs Creek, which, while this is an experimental facility here, it's more set up for production.
Yep.
So they're now taking some of those findings and operationalising them and moving into production for restocking. So, while we do the research here, they can focus on the production up there, which is really exciting. And that species is part of the program we call 10 and 10, which is about recovering 10 species of native fish, mussels or crays, not recovering, but creating more populations of those using conservation stocking to spread the risk of extinction. So if we've got 2 populations of an animal now, in 10 years, we'd like to have 3 or 4. 10 years after that, we might have 6 or 8. So eventually we're building up the robustness in the population and spreading risk from extinction. So pretty exciting.
Sounds really important. Like it sounds like a really good partnership between the delivery partners and the research to know what needs to be done and how to do it.
That's right. That's great. And a lot of that also, it builds on the work that's been done out of this place for the last 30, 40 years. We're now in a space where we can take that work and turn it into, direct recovery actions by doing these sort of things, which is really exciting. Awesome.
And what do we actually have in some of these tanks? Is there
Sticks and rocks.
sticks and rocks in this one? Wonderful.
About all we've got in here at the moment.
Do we have some crays?
Oh, yeah. I just went under a rock.
Okay. So more sticks and rocks. Oh, there's one there.
No, there's one under that. So that was.
Wait, I can do this one. Oh.
You can see that
Yeah.
under that rock. These are some crays that these are Murray crays, juvenile.
No, not that one.
Yeah,
That one? That one that counts?
There you go. Yep, that's good.
So these are juvenile Murray crays. No one's done much with Murray crays in a captive setting before. So these were hatched here in the ARI aquarium, and we're just looking at growth rates and different water quality parameters at the moment. And that's being done in conjunction with Snobs Creek hatchery. Really, Murray crays, are a really important species for a range of reasons, including recreational, but also culturally for some traditional owner groups. But the key thing we're trying to learn here is using this more common species as a proxy species for some of the really threatened species. So as part of the 10 in 10 program, we're going to bring South Gippsland spiny crays into Snobs Creek. No one knows anything about how to breed them or how to grow them. So if we can get some of that information on more common crays, that can be translated to the threatened crayfish. And also we've got the Glenelg spiny cray that's endangered and of real interest to people. So
it's a good test case that can be applied to where it's a higher risk if you've taken the captivity and things go wrong. So this is
a risk management thing, and it's a husbandry thing. So how to keep these things alive, basically, what works, what doesn't. And in particular, most of this work will be going to aquaculturists at Snobs Creek.
Yep.
They're set up to do. That's what they've trained to do. So, really important to get things going here. We had the mussels in some of these tanks here originally, and got them to breed. They're now taking the next step, with some of these species, which it's great.
Really cool. All right, should we head out to the boat shed? All right, let's do that.
While we're walking,
Yep.
I've got a question about VFA's expansion to Arcadia.
Yep.
Is there more scope for additional species to go there?
Yeah, there is. And we're having those, do you want me to answer here? Yeah, we're having those discussions at the moment. So Arcadia, the funding for that was largely around recreational species. So in particular, Murray cod and golden perch. But we're now having discussions with VFA management about broadening that scope for some of the warmer, smaller bodied but warm water species, but also species like catfish. So we're in active discussions about brood ponds for species like catfish. Yes. And the Snobs Creek hatchery, we've got our conservation component of it, but there's also, they're moving that to more a longer term focus on threatened species, which is great as well. It's great knowledge for fisheries.
Awesome. Cool.
Oh, so hopefully the internet is still working. We may drop out here, but we did test it the other week and it seemed to be good. So cross your fingers.
This is our gear shed, which we had built a few years ago. We used to keep all this gear off site, which was a real pain. Got a grant a few years ago to keep all this stuff on site, which has been fantastic.
Oh, look at this setup. That's some good help.
Along the walls that the other sections of ARI use as well. So there's all sorts of mammal traps and bird traps kept out here. But really the thing that takes up most of the room is boats for our work in aquatic ecosystems. Got a few different type of boats. We've just got punts on here, which are used for netting. They're used for radio tracking to get to sites. On the other side here, we've got electrofishing boats. For those who don't know about electrofishing, basically on a boat, we've got a big generator. The boat acts as a cathode, and there's things out the front of metal droppers act as an anode. We create an electric field which electrifies the water. The fish get stunned. They'll float to the top. We can pick them up. They're recovery bin on the boat. We'll weigh and measure them. Sometimes we'll tag them. I'll talk to you about tags in a sec. And then let them go. It's a really important part of the work we do because it's quite a safe, animal safe sampling technique. So there's been a lot of research on it over the years. Compared to netting and some of the other sampling techniques, it's quite safe on fish, and they recover really well. The reason it's important for government is because government spends a lot of time, effort and funds on restoring environments and restoring aquatic.
Yeah, aquatic environments and restoring waterways, it's huge as an environmental intervention in Victoria, even in Australia. It's plans like the Murray-Darling Basin Plan or Biodiversity Plan have got funding behind them to restore the environment, and water is such a big part of that. That's why it's really important that government can go back to the community and say, well, here's the impact of that intervention, whether it be water for the environment or planting riparian vegetation or flooding a wetland. That all costs. So it's important that government can go back and say, we did this and here's been the outcome. Yep. So that's one reason it's important. The other major component of the work we do is we try and work with managers to work out ways to do those interventions smarter. So I like to call it more frogs per drop or more fish per drop. But in the same way as
the marketer is always
right.
Well done.
In the same way as a farmer wants to get the best economic return for their water, their input, we want to get the best ecological return for our input. And so that's sort of falls back onto government. So how can you deliver water or deliver an intervention to get the best return on that? And I guess that's sort of, why a lot of people here in this game as public servants is to, make sure that the public get good value for
I mean, all those decisions are about trade-offs. And if you don't have clear understanding of what you're getting for a trade-off, it's very hard to make those decisions. That's a really good point.
I'll show you a few other things that aren't boats. So we've just got a couple of nets set up here. That's a pike net. They're quite a traditional way of catching aquatic animals. So in particular things like fish or turtles. These are a passive sampling technique that you sit in the water, basically the fish or the turtle or whatever it might be. Yabby will crawl into that net, sits on the bottom on the ground like that. Follow along with into a funnel and you then get captured. So it's a pretty traditional method. It's useful in some areas. When we capture the animals, there's a few different types of tags we can use. We've got radio tags here. I guess they're more pretty old school now, but they're still quite useful. So they emit a radio frequency. I've got one over here actually. I've got that turn on.
Oh,
So that's that tag beeping.
oh, that's so cool.
And what you can do is you can get a direction. So if I face away, you can hear that beep. I don't know if you can hear it still, but it's quite. But anyway, pretend it's getting louder. There we go. So you can get a direction on where the tag is, which is quite important if you're looking at habitats. That's radio tags. Historically, we used those a lot, still use them.
Oh, I recognise this one. This is an eel one.
Yeah, we're getting to that.
Oh, okay, all right.
Stick with me.
All right.
Come on. These are acoustic tags. The size of the tag's just indicative of the battery size. The technology, the receiver, a transmitter inside each of them is the same. It's just the battery size. So these let off a ping, which recorded on something like this, which is a logger station, which goes under the water, and it's a bit like a submarine ping. These record them. Just leave those in the water. So you set up arrays of these in rivers or wetlands or lakes. Got pit tags down here. There's sorts of things you might get put in your cat or dog, so you can take it, you know, if it gets lost and ends up with a pound, they can scan it. We use those on aquatic animals as well. These are external tags, which are mainly for people like anglers. If they catch a fish, they've got a unique code on them. They can ring up with that code. That information that they get is really important to us, and so then we can send them back information on that fish as well. So how much it's grown since we caught it, how far it's moved. And you can get some quite revealing information off those. Yes, that's a satellite tag, Andy. So these are going on eels now. So there's some work happening at the moment on longfin eels, and collaboration with Gunai. And some of these are popping up near Papua New Guinea at the moment, which is really exciting. They've got a fascinating life cycle. Eels, they live their adult life, juvenile and adult life in fresh water till they're 10, 20, up to 30 years old. They'll move down, then no one really knows what prompts it, why they would go at 10 or 30, but it's flow that prompts them. They'll move down into an estuary, then out into the ocean. They'll then travel up to between New Caledonia and Papua New Guinea, spawn and they die. Their larvae, which are called leptocephali, little leaf-like larvae that are clear, then drift down the East Australian current, or the Nemo current, as I like to call it.
EAC.
Yep, that's right. And back around into Victoria and southern New South Wales, where they're then prompted to go back up into rivers by flow in a particular year. So for being able to provide flow to attract those animals back in at the right time of year is really important. Speaking of flow, these are just flow meters, so we'll set them when we set our nets to work out, in short, work out how much water is filtered. So you can calculate that. We've been talking a lot about fish, but obviously the aquatic group does a heap of work on vegetation and birds as well. We just satellite tag some brolgers with these, looking at how they move around some wetlands in northern Victoria to inform management. These are water level loggers. We put those in wetlands to look at water level, which often managers don't have much information on. So how much water needs to be put in to get it to a certain level. It can be got by quite a simple little tool like that. They're all out in the field at the moment, but also got soil moisture probes. And that's really important because another question is when we're using water to try and make aquatic vegetation or riparian vegetation healthier, we need to know how far that water gets down into the soil profile. So we can put probes in the water and test how long water needs to sit there for to get to a level where it's useful for plants. So you might sit it there for 3 days and an extra day will get you, an extra 80% benefit.
Wow.
So it's trying to really
quantify that.
What else?
Is this fish sings?
That's sings.
Yeah.
That's dead fish. It's not actually dead fish, it's a mole. They don't do the dead fish anymore, but that's a trout cod, which quite iconic. One of those who are into aquatic environments. And this is backpack electrofisher, so as well as the big boats, the smaller boats, we've got backpacks for small streams. So they, instead of running off a generator, boats, they just run off a small battery, a 12-volt battery that sits in there, and then just a small net. Big animals up in the smaller streams. Awesome.
Really cool. Very Ghostbusters. I love the style.
Yeah. You should see the car that goes with it. Sirens. So it's nice.
Yeah. Thanks, Jared, for showing us around your wonderful shed. I think internet's going okay, or should we move back?
I think so. Let me just check the chat.
Well, I think this is the last stop of the tour. So what we might do is we might finish up the video component and we might jump online to answer any specific questions. Unless there's any.
I'm going to show them the boats because they missed the boats because it froze.
Okay.
So let me
Go to
just,
the tangle, the electrodes. That's the cool bit.
So-
And if you want to see more about these boats, we do have a VR experience about electrofishing. So you can stand on the boat, see that in action, and see some of the researchers picking up the fish as well. So that will also be on showcase at the DEECA Expo on Wednesday, where Tess and I will be there. But you can also find it on YouTube with a lot of our other videos that explains about research.
So these are the anodes. These sit in the water. I've stunned the water for around sort of 3 meters for the bigger boats and about 1.5 meters for the smaller boats, just the size of the generator. Yeah, the fish are actually attracted to the electricity, and when they get close enough, they get stunned. So you scoop them up, and there's a whole big well board where we measure, tag it. If we're doing a tagging program, get them back into the water.
Is this the grasser? These ones?
No, this is just the Smith root unit. This Grassel, the Grassel that we're talking about is a boat that we co-developed with Hans Grassel in Germany about 10 years ago. That actually was the first electrofishing boat to be able to work in salt water. So back there, looks the same. It works in salt water. That was a bit of a revolution for electrofishing. Normally, when you're electrofishing a boat like this, because salt conducts electricity so well, the electricity, the power just dissipates, gone. Yep. So these boats don't work. I don't know how he solved that, but it worked. They solved that. So these boats now.
And
works in estuarine and salt water.
Do many other institutes across Australia have this capacity or fleet?
I think so. People are now buying the Grassel boats. We've got 2 of them, or the Grassel units, putting in their boats, which is great. We've got one of the largest fleets. I think New South Wales Fisheries might have a similar size fleet. Yep. So it's probably us and New South Wales Fisheries. Generally, the government research agencies have the largest fleets. And then there's a couple of unis or some consultancies who have 1 or 2 boats of their own. Yep. But yeah, in terms of fleets, it's one of the bigger ones in Australia.
Yeah. And recently, South Australia called for your help, I heard.
They did, yes. They had a couple of their boats break down, and so we were able to pivot into action pretty quickly and lend them one of ours, which was really, we get along with the other state agencies really well, and all of the consultants as well. So we're happy to help them out with the boat so they get their work done. It's important work they were looking at river management and biodiversity data, keeping those data sets going. While it can be a pain in the year while you're doing it, 20 years back, people look and say, thank God they kept that data going, because we've now got something to refer to.
Yeah.
So,
well, it's a long-term data set, it's valuable. Yeah. Hard to see when you're doing it, but you get to look back. It's rewarding. All right. Well, we'll leave the tour there. Thank you so much for coming along and hearing about what we do, hearing about from our staff, the capabilities and expertise, and also just how passionate they are about the work they do. I think it's a really, really great opportunity to form partnerships in future and work out where we have the knowledge to support your needs, but also working out how we can share these science stories more broadly. So each of you who attended today, you are now an advocate for ARI in so many different ways. And thank you for coming along. Much appreciated.